Okay, so in this lab 6, we're going to talk about chemical and physical processes of digestion. So we're going to look at digestion of various molecules by digestive enzymes in these four activities. And so this is all accessible on PhysioEx. You guys have used PhysioEx before um, in one of the previous labs, and so you can access this through Mastering A&P. And so when you get to your study area, you can just see exercise 8 here for chemical and physical processes of digestion. And so just a few introduction words of introduction here. So we're going to look at enzymes. Enzymes are biological catalysts. And so catalyst means that they speed up chemical reactions, but an enzyme will not be used up in the reaction. An enzyme will just increase the rate of the reaction, but then it can catalyze multiple rounds of the reaction without being used up. Um, and so digestive enzymes are mostly known as hydrolases because they are able to hydrolyze or break down organic molecules into smaller building blocks called monomers. And this happens with the help of water that is being added to these monomers when chemical bonds are being broken down. And so, because this uses water, you have this hydro part in the hydrolases. Um, Hydrolases, as any enzymes, are very highly specific. So, a particular enzyme is only going to hydrolyze or digest or break down a specific molecule or specific type of molecule, or maybe a small group of similar molecules. The molecules that are being broken down are known as substrate molecules. So substrates are going to be molecules like proteins and carbohydrates and fats that are being broken down. Um, and this produces products, which are monomers in these reactions. Um, and so again, each enzyme will specifically recognize a particular substrate. For example, amylase will hydrolyze starch, but it will not hydrolyze cellulose that is also carbohydrate, but it has a different chemical structure. Also, enzymes require a specific environment to be active, and so changes in things like pH or temperature will make an enzyme inactive. Um, and so for human enzymes, all human digestive enzymes have the same optimal temperature, which is 37 degrees, and this is human body temperature. Um, and this is for obvious reasons because they are all inside of the human body. But the pH in different areas of the gastrointestinal tract will be different. And so, for example, in oral cavity, it will be neutral or around 7. But in the stomach, it could be highly acidic, around 2. Whereas in the small and large intestine, the pH will be almost neutral, but slightly acidic, so maybe around 6 or 6.5. And so looking at the first activity, we're going to look at digestion of starch by an enzyme called salivary amylase. So this happens obviously in saliva in the mouth. So what you guys will do in the lab is just work through this activity. So you will read about all of these objectives in the introduction and then do the experiment. There are basically two tests that you will be using to look at the action of salivary amylase. One is called Lugol's iodine or IKI test, and this is able to detect starch. There will be a color change in the presence of starch. The other test is called Benedict's reagent assay, or just Benedict's test. And this assay is for maltose, which is a sugar that is produced as a result of digestion of starch. So you go from starch to maltose. And with this starch or Legal's iodine test, you can detect the presence of the substrate, and then with the Benedict's reagents test, you can detect the presence of the product. So all of this is going to be very nicely described in this video. So here's a link. So you should watch this, and this actually shows you how this is done. And then once you have this idea, you can go through the lab and make sure that you submit your answers to pre-lab quiz. There's also going to be post-lab quiz and some control questions. And so make sure that you always submit these answers. And also make sure that uh, where when you are in the lab that you record. So there's going to be a button here says record data. So make sure you record the data when required. So up here during the lab, you will have instructions on what to do with these tubes, what to do with these reagents, and in the assay cabinet, at a certain point during the lab, you will have access to Lugol's iodine and Benedict's reagents to do your experiments. Um, and so this simulation basically recreates what you would have done in a wet lab. You know, if we had a wet lab, it actually recreates it pretty well. These are pretty much the same experiments. Um, so, so again, I don't want to talk about the results because you guys will get your results. So you will do these different conditions. As you can see, you will look at the influence of temperature on enzyme activity. So some of these enzymes will be boiled, some will be frozen. You will assay as you can see, so you will assay starch, the digestion of starch. Some of these, as you can see, are controls. So controls are conditions with known outcome that are used as a way to validate the experiment. So for example, here you have tube number 4, which does not have starch, just has water instead of starch. Um, and this is used to show, for example, that amylase alone will not be able to produce color changes, or buffer will not produce any color changes in these two tests. And so think about the value of these controls, and we will talk more about these different controls during the our virtual lab meeting, um, and um, and discuss why we need each of these controls. So when you're done with the lab, make sure that you record everything and you save your lab report that will be generated by the website. And so you just save that as a PDF. And so this is how. So you click on this printable version and print. So you select destination and save as PDF. So this is done for Chrome browser, but if you have a different browser, this may look different. But make sure you save this as a PDF, and then you can upload these PDFs to EasyLearn. So you will have to do this separately for every activity. Um, and then second activity, we will look at the substrate specificity for amylase. So remember that enzymes are highly specific. And so in this setup, you have amylase, but you also have a different enzyme called peptidase, and you have different substrates. So you have starch, but you also have cellulose, which is a different type of carbohydrate. You also have here some digestive bacteria. And we're going to assume that these digestive bacteria came from an animal, a ruminant animal such as a cow, where they could be used to digest cellulose. And this is an interesting example of how humans do not have uh, the type of bacteria that digest cellulose. So we cannot eat grass. We have to we cannot digest it. But many animals like cows actually have enzymes in their gut that allow them to digest cellulose. And so humans have many other kinds of bacteria. So we have trillions of bacteria in our gut, but just not the ones that allow us to digest cellulose and produce those enzymes. So again, go through this lab, through this lab activity, and you will have these results. So as you can see, you're testing starch and cellulose with amylase or peptidase. You're also testing bacteria could produce digestive enzymes. So, so again, in the end, you're going to save your lab report and then go to the next activity, which is looking at digestion of proteins. And so this is done with the use of this reagent called BAPNA here. And so BAPNA is a synthetic reagent, and it combines an amino acid um, uh, and that is shown here. So this is an amino acid, and then it also combines this yellow dye. And so when this is all linked together, it's colorless. But when an enzyme such as pepsin digests this and breaks this, what's known as the peptide bond here, you now have an amino acid and a yellow dye separately. And now this dye is going to show yellow color. Um, and so again, so you're going to do this and you're going to use a device called spectrophotometer to measure the color changes in your different reactions. And you can see there are three buffers here. So buffers are used to maintain different pH levels. And so you're going to assay the uh, what is the optimal pH for this particular enzyme. Again, you will get your results here, and you'll will boil one of the tubes, but then and then use different pH levels in these different tubes. Um, and again, make sure you save the PDF at the end. So in the last activity, you're going to look at digestions of fat with an enzyme called lipase. And so fats and oils, which are collectively known as lipids, are hydrolyzed to their monomers called monoglycerides. And these are, for example, like glycerol and also fatty acids. And so, because of these fatty acids, the hydrolysis of lipids such as fats or oils will produce acidic environment. Um, and so, these enzymes are produced in the pancreas. And the activity of lipases could be assayed using a pH meter. So this is just simply a device that allows you to measure pH. So if there is a production of fatty acids as a result of fat hydrolysis or oil hydrolysis, you're going to have an acidic solution. And so that will have a lower pH level than the one you started with, and the pH meter can detect that. And so if there is no digestion, the pH will remain the same as what you started with. Also involved in this is a substance called bile. And so bile are just inorganic salts. So these are not enzymes, but these are inorganic molecules that bind fats and they emulsify fats. So emulsification means that they produce droplets, like smaller droplets instead of larger globules or larger chunks of fats. And these smaller droplets will have a larger surface area that is accessible for lipases. And so bile is produced in the liver, and it is not an enzyme. But keep in mind that bile could increase pH, so this could actually mask the pH meter reading a little bit. So just keep that in mind when you're doing the experiment. And so you can observe bile action by adding bile salts. And so for this, as a substrate for this reaction, we're going to use vegetable oil. And so you have some lipase here, some bile salts, and then again buffers at different pHs. So remember that pancreatic duct empties into the small intestine, so the pH there should be more closer to 7 rather than 2 or 9. And so you will study this in under different conditions. And so just to note, for all of these reactions, the actual reaction would have taken 60 minutes to do, but in this virtual lab, this is compressed to 10 seconds. And so again, in the end, this will be recorded, um, and that will be our lab. And so we will meet in the morning at our regular time, and we will talk about the details of this a bit more as you guys are doing the lab. So make sure that you have access to your mastering. Make sure you have access uh, to EasyLearn, and we will do this over Zoom. So make sure you have that registered and running. So I'll see everybody in the lab.